In 2006, a team of archaeologists crawled into a cave called La Braña Arintero, tucked high in the Cantabrian Mountains of northwest Spain in the province of León. The cave was cold, dark, and sealed by centuries of sediment that had kept its interior remarkably stable, the kind of environment where organic material, given enough luck, does not simply rot into nothing. What they found inside were bones, 2 sets of them. 2 men who had died approximately 7,000 years ago and been preserved, not through any human intention, but through the accident of geology and moisture and darkness. The skeletons were given names from the site, La Braña I and La Braña II. La Braña I was the more complete of the two, his bones intact enough to suggest a picture of a man who had lived a hunter-gatherer life in Mesolithic Europe, who had eaten meat and fish and wild plants, who had left behind no pottery, no evidence of agriculture, and no permanent structure. For years after the discovery, researchers attempted to extract DNA from the remains. Bone degradation, contamination, and the limitations of available technology made it difficult. The breakthrough came in 2014, when Carlos Lalueza Fox of the Spanish National Research Council, working with collaborators at the Center for Geogenetics in Copenhagen, finally published a complete genome sequence of La Braña I in Nature. It was the first fully sequenced genome of a European hunter-gatherer ever obtained, and what it showed made headlines around the world and quietly shattered an assumption that science had held for generations. This hunter who had stalked deer and gathered plants in Mesolithic Spain 40 centuries before the Roman Empire existed, had dark skin. Not olive, not tan. Dark. African versions of the pigmentation genes, as Lalueza Fox put it in the research release. But he also had something else. He had blue eyes. And buried inside his genome, the same researchers found variants in genes associated with freckles. In a man with dark skin, in a cave in Spain, 7,000 years ago. If that sentence does not stop you, nothing will. Because what it means is that freckle S, those small scatterings of pigment that we have spent centuries associating with pale Irish skin and Celtic heritage and Northern European ancestry, are not where we think they come from. They are not a recent adaptation. They are not a product of the climate of the British Isles. They are a genetic inheritance from people who had been living in Europe for tens of thousands of years before anyone called it Europe. People who looked nothing like the populations we now most associate with freckles. People whose entire world vanished and whose languages are gone and whose gods have no temples, but who left something behind in the genomes of living human beings that still shows up right now, today, on the faces of children in Cork and Edinburgh and Oslo when the summer sun comes out. That is not poetry. That is molecular biology, and the story it is telling is one of the most extraordinary pieces of human history ever extracted from ancient bone. To understand it fully, you need to understand 3 things. You need to understand what the MC1R gene actually does. You need to understand who the first Europeans actually were. And you need to understand the 3 waves of migration that replaced again and again the people who came before, without ever fully erasing what those people left behind. When you put all 3 of those things together, freckles stop being a minor cosmetic detail and become a direct signal, a biological radio transmission from a pre-agricultural world, still broadcasting. Start with a gene, the MC1R gene. Melanocortin, protoactive 1 receptor, sits on chromosome 16q24.3 and consists of 951 coding nucleotides. It is a small gene. It is 1 exon, and it is one of the most polymorphic genes in the entire human genome, meaning it varies more between individuals than almost any other gene we know of. What it does, at the cellular level, is control which type of melanin your skin produces. When it is working at full capacity, UV radiation triggers the release of a hormone called alpha-melanocyte-stimulating hormone, which binds to the MC1R receptor on your melanocytes, the pigment-producing cells, and triggers the production of eumelanin. Eumelanin is dark brown to black. It distributes evenly across the skin surface. It acts as a natural sunscreen, absorbing UV radiation before it can damage DNA. It is why people with fully functional MC1R can tan. When UV hits them, the system activates, the eumelanin increases, and the skin darkens as a protective response. When the MC1R gene carries certain variants, and the scientific literature identifies multiple specific ones, including ARG151 size, ARG160TRP, and ASP294 highs, which appear with exceptional frequency in Irish populations, the signal chain is disrupted. The receptor does not respond efficiently to the hormone. The melanocytes switch their production toward pheomelanin instead. Pheomelanin is reddish yellow. It does not provide meaningful UV protection. And critically, it does not distribute evenly across the skin surface. It concentrates. It pools in small clusters in specific cells, producing the uneven pigmentation we call freckles. A 1998 study published in the American Journal of Human Genetics, sampling the Irish population specifically, found that 75% of Irish individuals carried at least one MC1R variant and 30% carried two. These were not rare variants, they were the norm in that population. The specific variants most strongly associated with both freckles and red hair, the ARG151CIs and ASP294-His variants, were significantly more common in Irish individuals who freckled across multiple body sites, including face, neck, shoulders, upper limbs, back, and chest. A 2017 study published in Forensic Science International, examining 458 individuals from Spain, identified 4 genetic loci associated with freckling: MC1R, IRF4, ASIP, and BNC2. Across European populations today, contemporary research estimates that between 60 and 80% of people in Ireland, Scotland, and England carry at least one reduced function MC1R variant, a frequency that is extraordinary by global standards. East Asian, South Asian, and Sub-Saharan African populations carry these specific high penetrance variants at dramatically lower rates. The variants are European. They are most concentrated in the northwest of Europe, and they are, as the ancient DNA now tells us, far older than the populations currently most associated with them. Here is where the 3 waves begin. The first humans to establish themselves across Europe after the last ice age are called Western hunter-gatherers, or WHGs. They expanded across the continent roughly 14,000 years ago, following the retreat of the glaciers, moving from southwestern refugia northward and eastward, colonizing a landscape that was transforming from tundra to forest. They were the dominant human population of Europe for thousands of years. And when geneticists began extracting their DNA from skeletal remains found across the continent, from Spain, from Luxembourg, from Hungary, from Britain, what they found was consistent and startling. The WHGs did not have the light skin genes now common in modern Europeans. They lacked the derived variants at SLC24A5 and SLC45A2, which are the primary genes responsible for the pale skin characteristic of modern Northern Europeans. Their skin was dark, but they had blue eyes, often, and they carried MC1R variants at frequencies consistent with a population prone to freckles. Labranja 1 from Spain, sequenced in 2014. Then came Cheddar Man. He was found in 1903, when workmen excavating Gough's Cave in the Cheddar Gorge of Somerset, England, broke through into a chamber and discovered a human skull. The skeleton turned out to be the oldest near-complete human skeleton ever found in Britain, dated to approximately 10,000 years ago, making him a contemporary of the post-glacial landscape, when Britain was still connected to continental Europe by a land bridge. For most of the 20th century, Cheddar Man was reconstructed and displayed with light skin and brown eyes, the assumptions of the era plastered onto his bones without any genetic evidence to support them. Then, in 2018, a team from University College London and the Natural History Museum drilled a 2mm hole into his skull and extracted bone powder for DNA analysis. Researchers, Professor Mark Thomas and Dr. Johan Diekman, led the genome sequencing. What they found dismantled the old reconstruction entirely. Cheddar Man had blue eyes, dark curly hair, and skin color described by the scientific team as dark to black, with a statistical probability of approximately 76% for that determination based on the pigmentation gene variants present in his genome. Chris Stringer, the Natural History Museum's research leader in human origins, who had spent a decade analyzing the bones found in that cave, said that the combination was not surprising to ancient DNA geneticists. It was typical. The dark-skinned, blue-eyed Cheddar Man was not an anomaly. He was the representative European of his era. His tribe's genetic signature can be traced to individuals found in modern-day Spain, Hungary, and Luxembourg, confirming that this physical profile extended across the entire Western hunter-gatherer range. Around 10% of indigenous British ancestry today can be linked to Cheddar Man's people, according to the UCL researchers, a surviving fragment of a population that was otherwise almost completely replaced by the waves of migration that followed. These people occupied Europe for thousands of years, hunting, fishing, living in small, mobile bands, leaving almost no permanent structures, which is part of why their existence was so invisible to history until ancient DNA revealed it. But then, approximately 9,000 years ago, the first wave of replacement began. From Anatolia, what is now Turkey, a population of early farmers began moving westward into Europe. These people carried agriculture, wheat, barley, sheep, cattle, pottery, and they carried different genes. They had lighter skin than the WHGs, driven by selection at those SLC24A5 and SLC45A2 loci. They had predominantly darker eyes, and they did not carry the MC1R freckle, associated variants at the same frequency as the WHGs they encountered. As they moved across Europe, they mixed with WHG populations to varying degrees, and in many regions replaced them substantially. The WHG genetic signal diluted across most of the continent. The MC1R variants that had been common in WHGs became less frequent in the mixed population that followed. And then the second wave. Approximately 5,000 years ago, from the Pontic-Caspian steppe, the vast grasslands north of the Black Sea that stretched through what is now Ukraine, southern Russia, and Kazakhstan, a third founding population of Europe began moving westward. These are the people geneticists call the Yamnaya, named for the Yamnaya archaeological culture, identified from burial sites across the Ukrainian and Russian steppes. Their signature was the kurgan, a burial mound built over a wooden chamber containing the body of a single individual, often accompanied by cattle bones, ochre pigment, and copper ornaments. These were not random burials. They were monuments to hierarchy, to power concentrated in specific male lineages. The Yamnaya had mastered two technologies that changed the physics of human migration: the domesticated horse and the wheeled wagon. With horses, they could cover distances that foot-traveling farmers could not. With wagons, they could carry enough supplies to sustain movement across open steppe for months at a time. They were pastoralists, their wealth was in cattle and horses, their economy was mobile, and their capacity for rapid movement across a landscape was unprecedented in human history to that point. David Reich of Harvard Medical School, whose laboratory has produced some of the defining ancient DNA research of the past decade, has described what the Yamnaya did to European populations in terms that are almost impossible to absorb. Speaking in 2025, he stated that in Britain there was a 90% plus population replacement within decades. His colleague and archaeologist Barry Cunliffe stated that no one, not even archaeologists in their wildest dreams, had expected such a high steppe genetic content in the populations of Northern Europe in the third millennium before the Common Era. Reich's team reported in 2015 that even today, DNA from modern Western, Central, and Northern Europeans carries close to 50% genetic contribution from the Yamnaya and their descendants. 90% replacement at the leading edge of the migration, 50% surviving genetic contribution today. Those two numbers together describe a demographic event with no peacetime parallel in documented human history. The Yamnaya and their related cultures, the corded ware people who carried that steppe ancestry into Central and Northern Europe. Asti moved through a continent and rewrote it. They did not carry the MC1R freckle-associated variants at significant frequency. They were not a freckled population. When they replaced between 70 and 90% of the genetic ancestry of country after country across Europe, the expectation might be that those variants, already diluted from WHG levels by the Anatolian farmer migration, would be reduced to near zero in the populations that resulted. And across most of Europe, that is roughly what happened. But not in the Northwest. Not in Ireland, not in Scotland, not in Wales, not on the Atlantic fringe. These were the areas that the steppe migration reached last and least completely. They were geographically buffered by the Channel, by the Irish Sea, by the mountains of the western Atlantic facade. The replacement happened, but it was attenuated. The WHG ancestry that had survived the Anatolian farmer arrival survived the steppe arrival too, at higher proportions than anywhere else on the continent, and embedded in that WHG ancestry, carried forward through 10,000 years and three demographic events that each should have extinguished it, were the MC1R variants, the freckle genes, still present, still expressing, still showing up on the faces of people whose distant ancestors had looked nothing like them, but had passed them something durable through every generation in between. A 2023 study published in Nature, the most comprehensive ancient European genome analysis ever conducted at that time, covering 356 ancient hunter-gatherer genomes spanning 30,000 years, led by Cosimo Post at the University of Tübingen, and including researchers from the Max Planck Institute for Evolutionary Anthropology and 15 other institutions, confirmed this trajectory in molecular detail. It showed the WHG ancestry component surviving in diminishing but persistent frequencies across successive demographic events, with its highest modern residual concentrated in Atlantic and northwestern Europe. The PNAS study on pigmentation evolution in West Eurasia, published in 2020, added a critical layer. The researchers found no evidence of strong positive selection driving MC1R variants in European populations. They were not selected for, they simply persisted. They survived not because they were advantageous, but because they were not sufficiently disadvantageous to be selected out, and because the populations that carried them at highest frequency happened to occupy the edge of the continent where each successive wave of replacement lost momentum before it could complete its work. That finding matters enormously. It means freckles are not an adaptation. They are a survival. They are the biological residue of a people who could not outrun the demographic forces that moved across their world, but whose genome, in fragments, in partial inheritance, and the specific variants on a specific gene, outlasted everything else those people built or believed or became. Before we reach the medical consequences of carrying these ancient variants, it is worth pausing on something that the science does not address, but that the historical record makes vivid: the extraordinary instability of how freckles have been perceived across the centuries they have been visible. For most of recorded European history, freckles were considered a blemish. Medieval European medical texts treated them as a skin condition requiring remedy. By the Victorian era, the cosmetic industry was actively marketing treatments to eliminate them. Arsenic-based creams, lemon juice applications, and chemical preparations sold specifically to women who wanted the fashionably pale, unmarked complexion that Victorian beauty culture demanded. The irony is complete. The same Atlantic fringe populations that retained the highest WHG ancestry, and therefore the highest freckle frequency, were also the populations that Western European beauty standards most pressured to erase the trait. It was not until the mid-20th century, and then more fully in recent decades, that freckles began to be reframed, first as a marker of youthful charm, then as a feature to be celebrated, and now as something people who were not born with them attempt to replicate cosmetically. The genetic legacy of Mesolithic hunter-gatherers has, in the space of a century, moved from something women tried to bleach off their faces to something others tried to paint on. The gene itself was indifferent to all of it. The biology of carrying these ancient variants, however, has consequences that are not cosmetic. Loss of function, or reduced function. MC1R variants blunt the eumelanin response, resulting in lower eumelanin and higher pheomelanin production, which manifests as red hair, fair skin, freckles, and poor tanning. Beyond pigment, MC1R signaling influences DNA repair responses, oxidative stress handling, and cell cycle control following UV radiation, which means variants can raise skin cancer risk in ways that are not fully explained by pigmentation alone. Research published in the Proceedings of the National Academy of Sciences demonstrated that pheomelanin is not merely a weaker sunscreen than eumelanin. It is actively photounstable and has been shown to amplify ultraviolet A-induced reactive oxygen species. That means freckled skin does not just tan less effectively. It generates additional molecular damage under UV exposure through a mechanism entirely separate from its failure to produce eumelanin. A landmark study from the Wellcome Trust Sanger Institute demonstrated that a single MC1R variant can increase an individual's melanoma mutation burden by an amount equivalent to approximately 21 years of additional sun exposure, meaning a 30-year-old with one MC1R variant may carry the UV damage profile of a 51-year-old without one. Individuals with fair skin and red hair, driven by MC1R loss of function, exhibit the highest risk for developing melanoma globally. The ancient gene that produced freckles in a dark-skinned hunter-gatherer 7,000 years ago now creates measurable, quantifiable medical risk in its current carriers. A consequence the people who first carried it could never have anticipated, living in the low UV environment of Ice Age Atlantic Europe, in forests so dense that direct sunlight was itself intermittent and scarce. There's a retired schoolteacher named Adrian Target who lived, at the time of a remarkable 1997 study, approximately half a mile from Gough's Cave in Cheddar, Somerset. The study was straightforward in its design. Researchers extracted mitochondrial DNA from a molar tooth belonging to Cheddar Man, and then tested local residents, many of them connected to a school near the cave site, to see whether any living person shared that maternal genetic line. Mitochondrial DNA passes unchanged from mother to child, generation after generation, making it an extraordinarily precise tool for tracking maternal lineage across time. When the results came back, one match stood out. Adrian Target, a history teacher, carried a direct maternal genetic connection to a man who had died in a cave 10,000 years ago, less than a mile from where Target lived, worked, and taught. When told of the discovery, Target reportedly expressed a mixture of surprise and quiet pride. He had been teaching local history for years without knowing that the most local history of all was written in his own cells. The connection was later confirmed and extended by the 2018 full genome sequencing of Cheddar Man, which placed Target within the broader population that carries WHG maternal ancestry. 300 generations of people living in and around the same Somerset landscape, passing mitochondria from mother to child across a span of time that absorbed the entire Roman occupation of Britain, the Viking invasions, the Norman conquest, the Black Death, the Industrial Revolution, and every other event that layered itself over that land. A spy! And still the molecular thread held. Cheddar Man had dark skin and blue eyes. Target did not. But somewhere in Target, the original genome of the first Briton persisted, silently, in the one type of DNA that cannot be diluted by paternal ancestry. Some of that molecular persistence is on chromosome 16. Some of it is in the MC1R gene. So some of it expresses when sunlight hits the skin of a person who has no idea any of this happened, as a scatter of pigment across a nose and cheekbones. That Labranja hunter, Labranja 1, the man whose bones were found in a cold cave in the Cantabrian Mountains in 2006, lived in a world that is now completely gone. The forests he moved through were dominated by oak and hazel and elm, the Mesolithic woodland of Atlantic Iberia, dense enough that the landscape would be unrecognizable to anyone alive today. His band was small, perhaps 30, perhaps 50 people. They followed game and salmon runs and the seasonal patterns of plant food. They built nothing permanent. They left behind no writing, no monument that survived, no artifact that carried a name or a story. When the Anatolian farmers arrived and their descendants multiplied and the population dynamic shifted irreversibly, Labranja's people were absorbed, displaced, and ultimately replaced. Not in a single violent moment, but in the slow arithmetic of demographic advantage, generation by generation, until the genome signal of that entire human world was reduced to a whisper in the DNA of the populations that came after. What was left of him, of them, was not a monument. It was a gene variant. It was the disruption of a pigmentation pathway. It was the reason that pheomelanin pools in specific cells instead of spreading evenly, and the reason that sunlight, falling on skin that carries this variant, produces not a uniform darkening, but a constellation of small, concentrated pigment deposits on the face of someone who has never heard of Labranja and has no reason to think that the feature they see in their mirror every morning connects them to a vanished world. It does. Freckles are the last visible trace of the oldest Europeans, not a quirk, not a cosmetic accident. A message from people who had no way of knowing they were sending one, transmitted across 7,000 years of migrations and replacements and demographic upheaval that should, by any reasonable probability, have erased it entirely. It wasn't erased. It's still here. And now you know exactly where it came from.